All right, so I think one of the topics that's been mentioned most often in presentations until now and hasn't been presented by itself is the brain imaging data structure, standard for organizing and sharing and describing neuroimaging data and more. We're very fortunate to have Dora Hermes from Mayo Clinic here, who is will give a particular angle, I believe, on this from sort of her perspective in research, but is also a member of the steering group of the brain imaging data structure community, and so has kind of like a very broad overview as well as her very specific sort of angle, and has made like extensive contributions to BIDS over the years, and so we're really fortunate to have her and present about this. Go ahead.
Thank you very much for having me here this week, to the organizers. It's been really wonderful to be here. I spent a little bit of time in Seattle during my PhD, and it's always really wonderful to be back and get to talk about interesting data for a week. So it's wonderful. Just as a note, after my talk, I have a conversation with my program officer at 10 o'clock, so I'll be rushing out, but I'll be here for lunch, and I'm happy to chat about BIDS or multimodal data, etc., for the rest of the day. So it's just that after my talk, I'll be rushing out for a little bit. I would like to talk a little bit about BIDS today, and as Ariel mentioned, I kind of give this always my own angle, because there's a reason why we started working with BIDS in particular, and very, very practical reasons. And I think it's important to emphasize that we're not just structuring our data in a certain way just because it's easy for other people to share the data, or we have to. It's actually just practical solutions to problems that we have. And so in my lab is based on Mayo Clinic. We're doing a lot of work that is fundamental insight into how brain networks are driven by electrostimulation or vision. And the reason why we're at Mayo Clinic is because brain stimulation therapies are rapidly advancing. And neurosurgeons are implanting electrodes, for example, in structures to treat depression, movement disorders, epilepsy, or visual prosthetics. And in order for those types of therapies to be able to move forward, we need to understand networks. Because the stimulation, when we stimulate, for example, a single site in hippocampus, it doesn't just stay there. It actually propagates throughout the network and drives both direct connections and indirect connections. And the way in which you understand those connections is, of course, not by implanting electrodes throughout the brain. There's only about, we can implant about 256 electrodes, which may seem like a lot. But in terms of the size of the brain, that's actually very sparse sampling. They're generally implanted only on one hemisphere, for example. So we need non-invasive neuroimaging techniques to get a better view of human brain networks. You can also do these types of non-invasive techniques in large groups of healthy subjects to understand how different areas are connected, to map development, to track how different diseases would affect networks and where these devices would have to be implanted to optimally treat network diseases. And so when we stimulate, for example, an area and we measure other areas, it's actually interesting because if we go back to 1966, this was actually a technique to map networks even before diffusion imaging existed. And so this is research by Otto Creutzfeldt, the son of actually the Creutzfeldt from Creutzfeldt-Jacobs disease. They stimulated the thalamus and they measured evoked potentials in cat cortex. And they found that this early evoked potential, they measured that in cat cortex after stimulating the thalamus and could then use that potential to understand whether there was a lesion in the optic radiations or how the changes between whether there were any changes between the thalamus and the cortex. This is quite an old technique to use brain stimulation or brief stimulations to map networks. And it's also, if we think about stimulating, we can also stimulate the brain with vision, with visual inputs or with auditory inputs. And one of the most commonly used stimulation protocols is actually brainstem auditory evoked potentials. Almost every baby, when they're born, gets an EEG test with brainstem auditory evoked potentials to check their hearing. And when they stimulate with like a brief pulse, a brief auditory blip, and they measure these evoked potentials, you can see that all these little peaks in the auditory evoked potential have a name. And those have been linked to individual nodes in these network. So we can use evoked potentials if we understand the brain structure extremely well to specific nodes in the network. And these changes in these nodes are starting to be linked to brain tumors, hearing deficits, just a simple clog in the auditory canal, or changes in excitability. And so clinicians use this particular waveform to actually detect changes or disease-related changes. So if we can start to combine these types of structural measurements in cognitive networks with evoked potentials, we might get a much better view on how these circuits are changed in different types of diseases, and go from really start to use these things as biomarkers, electrophysiological biomarkers for networks. And so, for example, we can stimulate a part of, like, this is the cingulum bundle rendered in one of the human brains. And we can imagine we have an implanted electrode. We work with epilepsy patients who have electrodes implanted for clinical purposes. So we can stimulate one of these electrodes with a brief pulse. Clinician is neurologist is in the room to make sure that we don't induce any seizures. We stay below the safety threshold. And then we assume that these signals spread throughout the white matter bundles in different directions. And if we have an electrode placed in one of the other areas, for example, here, we can then, after, say, 30, 40 milliseconds, we can measure an evoked response when the signal gets there. And so we can track signals throughout these white matter bundles. And so what I would like to share today is how we integrate these types of evoked potential data, how we share them in the brain imaging data structure, and integrate those, for example, with diffusion MRI atlases to quantify the speed of neural signal propagation across development, or how we basically integrate these measurements with diffusion MRI in the same subject to to better understand direct and indirect connectivity in the human limbic system. And then I would like to do a demo because we've shared with all these publications, we share data in the brain imaging data structure on OpenNeuro. And so if you want to work with those data, I want to show you how to load those and how to think about those data and how to basically easily access those for other purposes, because these are just a few of the questions that you can ask or address with these data. And so one of the things that is well known from diffusion imaging is that these types of networks that we get from diffusion imaging develop throughout the lifespan. This has been extremely robustly mapped with for example, these in the paper, for example, by Jason Yeagman in Nature Communications in 2014. But many other studies have shown that these networks evolve throughout the lifespan. And if we look at, for example, the inferior longitudinal fasciculus, those changes in the white matter structure that are mapped here with automated fiber quantification and quantitative MRI, those often show a long developmental trajectory where these tracks develop until the age of 30 to 40 years old. And so we see that these structural connections develop for a long time. But when we think about the speed with which the neural signals travel, we actually don't know whether these measurements would have any prediction of that. Like with that, whether these non-invasive measurements from diffusion imaging, are those actually indicative of changes in the structure that affects neural signal propagation, or is this more the support structure around it that's still developing? And we would think about, if we think about that, it's actually, you think like, maybe this has been resolved in animal research. We already know this, right? But that's actually not the case, because human brains have a much longer myelination compared to many other primates. If you look here at the image on the left, you see the years, and this is a human brain where you see the myelinated fiber length density. And you see that these humans have this additional growth in myelin after the age of 20 years old. Whereas in chimpanzee, it actually plateaus after about 10 years of age. And they don't have this additional growth spurt in terms of brain development. So humans have a unique brain in terms of the connectivity. So in order to get these types of measurements and understand how the structure influences the signal propagation and thus the brain function, we have to actually do measurements in humans. And so if we think about this question, like what's a typical transmission speed across the arcuate fasciculus, it can be anything from 2 meters per second, like a person walking, to a peregrine falcon flying over 100 meters per second, because these are all realistic transmission speeds given the axon diameters, which is linearly related with the speed with which things travel, the speed with which the signals travel across the axons. So of course, we could measure postmortem axon diameters in these structures, but we can also use the single pulse stimulation as I described. And since we have the opportunity to work with these types of patients, we can stimulate these electrodes with a brief pulse, 200 microseconds in duration, and then we can measure the evoked potentials in other structures. And the responses are extremely robust. Here you see a set of individual trials, and they all have these like early evoked waveforms. And we can average those, and then we can detect, for example, an early positive or negative peak for one simulation pair and start estimating the speed or the delay between two sites. So we can estimate the transmission delay. And if we look at those data from 2 patients, you can see here that if you see the dashed line, you see this early negative evoked potential measured on the brain surface, which has been related to direct cortical connectivity in a 38-year-old in the dashed line, and in a 4-year-old in the solid line. And this is where I think it's important to switch to BIDS for a little bit and talk about how we could then do these types of studies in large groups of subjects, because you see here that we did this study in 74 patients. Now, for intracranial EEG, each center, large centers, see about 10 to 15 patients a year. And so to get 74 patients, you'd have to either wait for a long time or have a larger center. And so the question is really for these types of studies is how do you scale this with each center only having a few patients a year? A neurologist has to be present also during stimulation. So you have to have people who are invested in this. And so the way in which we do this in our studies is we have a very large center at Mayo Clinic. They see about 40 to 50 patients a year. It's one of the largest centers in the world. And we have very invested clinical collaborators, both from neurosurgery and from neurology. This, for example, Greg Worrell, Nick Gregg, and Brian Lundstrom are some of the wonderful neurologists. Frans Leijten is a neurologist at the UMC Utrecht. They've done these types of studies for the last 10 to 15 years from a clinical perspective. And we have standardized data collection and collaborations with Washington University in St. Louis. And so when we ask about how to scale, we have multiple centers, among which centers who have done this for a long time, such as a center in the Netherlands. Harmonized data collection using standardized reporting equipment in the same ways as you have standardized scanner protocols. And neurologists who are invested in this type of research and are interested in this from a clinical perspective to ensure safety. But even if you do all that, you still have to get the data in the same structure and harmonize the data curation aspect of this. Because in addition to just a data file, you also need electrode positions, for example. You also need the MRI to be in the same space. You need to have these all co-registered and matched up, because otherwise you cannot integrate these data across centers. And so that's where we just ran into this question, like, how do we do this? How do we go back and get all the data from the last 15 years from Utrecht, the UMC Utrecht, integrated in one format? And that's where BIDS is just extremely practical, because for fMRI, for example, the BIDS structure was already there when we started these types of projects. And it's the nice thing is that it's human, not only machine readable, but also human readable. You can read all the files. So if I give one of my TSV files that has one of the metadata files to one of the research techs and ask them, can you annotate the seizure onset zone? they can just open it in Excel. They can add a note on what's the seizure onset zone. They don't need to learn anything new. I cannot start teaching them a new program like an XML. That doesn't work in the clinical work. They're not going to give you the seizure onset zones. They're going to give it to you in a separate file that you then have to put in, which increases the risk for error. And so it's really important that these files are not only human readable, but in addition to that, I can then import it in MATLAB, Python, or whichever GUI we use or like tool we use, and it's machine readable as well. And we can check whether they imported the correct phrases. We can do checks, etc. And so when we started this project, before we started working on the IEG, I talked to Chris Gowrolowski at Stanford, and we were like, okay, can we just use this for IEG as well? And he said, like, yeah, this is actually a community effort. So for anyone here in the room, if you want to start getting involved in this, BIDS is not driven by a few people. There's a lot of people who contribute, but there's also a large community involved, and they actually want new people to start, you know, to get involved in that. And everybody is excited about it when someone new contributes or makes a point about something that's necessary. And he told me, yeah, you can just, you know, there's an MEG bits extension. People are working on that. Why don't you just do the IEG bits extension? I was like, I can just do that. So yeah, you can just do it. And there's other people interested as well. And Chris Holdgraf from Berkeley is interested. So why don't you get together? So we were like, okay, we'll get a large group of people together. We emailed a bunch of collaborators and they all commented on our Google Doc. And we communicated closely with the EEG bits extension, such that we were extremely well aligned and all the fields were the same and everything was consistent. And we did things in a way that we can actually easily integrate EEG and IEG data in BIDS, combine them also, collect them simultaneously, but have all the necessary fields that are necessary for both data types. And there's other extensions, for example, multi-electrode array recordings, PET, CT, eye tracking, motion, other derivatives that I'll touch upon later. So there's a lot of opportunities here to actually get involved and with other people who are interested in large data sets and sharing it. The advantage, as I mentioned, is that it's all human and machine readable and validatable. So if you have a BIDS data set, you can run the BIDS validator and it tells you you're doing good, which is fun. It's available online, easy to run. And then when everything is BIDS compatible, you can load a time series data from a bit structure and you can actually visualize it with the onsets aligned, because you can annotate which visual stimuli or what type of electrical stimulation was applied at that time. And the same for if you have a compatible fMRI or MRI data set that might have the same events, they will be annotated in exactly the same fashion. We can also render the electrode position. So in terms of the video you saw in the beginning, that's all the reason why we're able to do those things is because the electrode positions, the MRIs, the diffusion imaging are all stored in BIDS. We know the spaces are labeled. We know which space it's in, and we have everything in the standardized bit structure. So if someone else comes into the lab, they can basically read and see what space it's in. So it also makes it much easier to communicate with people within the lab about how the data are structured, because they can also just look it up online. And so there's a lot of information on the BIDS website. We're working on getting this a little bit better organized and easier to browse. But if you have comments on that, please let us know. There's also the BIDS starter kit, which will help you get familiar with some of like, how do I structure data sets in BIDS? And as I mentioned, the BIDS validator, and feel free to post an issue or contribute. Don't be hesitant to put in a GitHub issue I don't understand this. This is not very clear. That's actually a very useful comment for the BIDS community, is to, if people say something that is not clear, because maybe, maybe it's not clear for a lot of people and it should be clarified. So if you want to get involved, there's a lot of extensions in BIDS. And just some examples of what my lab is working on. We're contributing to the generic BIDS connectivity schema, because as I showed you, we're trying to do these types of IEG connectivity matrices, but we should combine that with with DTI connectivity matrices or fMRI connectivity matrices to have, for example, this is a little bit small, but for example, undirected and directed graphs. How do you indicate the symmetry? How does all of this fit in the same schema, such that if we share our connectivity matrix, that other people can then integrate it with their data from a different modality or from a different study. We're working on electrophysiology derivatives and annotations. For example, how do we standardize artifacts? So should I say that something is bad? What does it mean? Is there noise in the signal? How do we annotate that? And there's currently a discussion ongoing on stimuli and how those should be annotated. For example, movies, if those are annotated in a standardized fashion, they're easier to read for other people. So this is one of the ongoing discussions, is like, should this be a BIDS extension or not? Is this a big project? Is this a small project? And so there's, you can see some of these ongoing discussions of like, where is BIDS going in terms of describing these things in a standardized fashion. And so for harmonizing the data curation across within and across centers, I mean, I say IEG here, but of course there is like a lot of other data types that we have for every patient. We have MRI data, we have CT data. Sometimes even MEG or EEG data are recorded in the same patients. BID seems to work practically. And so when we have that, we don't just have these 2 patients. Now we can start doing this across our 74 patients. We can detect this early response. And we can plot that. We can plot these responses that are just sort of traces in these colors with the larger negative with a larger negative amplitude in yellow and the detected peaks here in black. And you can see that the older patients all had faster responses compared to the youngest patients. And there was a significant negative correlation between age and these delays. And so if you plot that, for example, these conduction delays as a function of age in terms of the latency, you can see that also when we fit this with either a first or second order polynomial, you can see that there's that age explains about 43% of the variance in these delays. And this is not just the case. And in this case, I'm showing an example for the arcuate fasciculus when we're stimulating temporal and measuring frontal. We restricted this analysis to the areas where we had at least 20 subjects that had electrodes on both sites. Because of course we can only measure this connectivity if we have a subject that has electrodes both frontal and temporal. And there's variability in the subjects in terms of where the electrodes are placed. And so we see some patients with visual coverage, but not as much. In terms of the ECOG recordings that are done on the surface in this study, we generally report from one hemisphere, so we cannot map the, for example, the corpus callosum. So we looked at a certain number of endpoints from the Yay atlas that are endpoints from the arcuate fasciculus, the superior longitudinal fasciculus between frontal and parietal regions, the section of the superior longitudinal fasciculus that connects frontal to central regions. This is much shorter than that, so we wanted to separate that out. You can also see that the delays here are shorter than the delays here. But you can also see that you can still see that there's a similar trend with age. Everything decreases with age in terms of the conduction delays, and the temporal parietal Osnon tract. But now, if we actually want to think about these transmission speeds, what we actually had to calculate is the fiber length. And not all these subjects had an individual diffusion imaging scan because these data were collected 10 years ago. So we can't necessarily do a diffusion tractography in every individual subject. And so we had to estimate this from an atlas, and we could use the Yay atlas that's available here and move that to the individual subject space so that we have estimated fiber length, and so we could calculate conduction speeds. All the code related to this is available on GitHub, by the way, all the data are available on OpenNeuro. But if you're interested in reproducing any of these things, it's all available on GitHub. And then when we do this, we can actually see these conduction speeds increase with age, peak around 40 years. And we see that at a younger age, the brain signaling and the arcuate fasciculus goes about as fast as someone walking, around 2 meters per second. But around 40 years of age, we're a happy biker. So we go around 5 to 6 meters per second. And this is about twice as fast. So this is not as fast, for example, as you would, it's the corticospinal tract, which is much faster, much thicker axons. But what we have to keep in mind is that the brain has a limited space. So we cannot have all fibers be super thick because there just simply wouldn't be space for that. So there are some fibers, such as, for example, corticospinal connections to corpus callosum, that have much thicker axons and are much faster than the arcuate fasciculus. And we see this increase in speed for all the different tracks that we measured, either peaking around 40 to 50 years of age or just increasing. And we didn't have many older subjects, so I don't necessarily take this. We don't know what happens when we get even older than that, because patients often don't get implanted with electrodes after this age. And so the last question that I just mentioned, like one of the things I just mentioned, is that the brain is not a mass of fibers that all have the same axon. The axon diameters differ between less than 1 micrometer to up above 10 micrometers. And so U fibers are smaller than these large association tracks. And so if we look at these types of tracks, like for example, frontal U fibers, parietal U fibers, and central U fibers, we see, however, that these all follow, the conduction delays also follow a similar trajectory, but they were overall slower when we calculate the speeds. This only went up to like 2 meters per second, whereas these large fibers actually went up to like 4 to 8 meters per second. So we see here that these are, but these develop in a similar way. So the entire brain seems to be developing for a long time. So when we integrate these intracranial data with the MRI atlases, we can see that the adult transmission speeds are about 2 times faster than kids, and that this developmental trajectory is comparable to that measured with diffusion MRI. And so these diffusion MRI measurements may actually tell us something about the microstructural changes such in myelin and axon diameter, and those do affect brain function, transmission speeds. And U fibers are slower than large cortical connections. And one of the things that we also noted in the U fibers is that the sensory motor connections were matured or had a faster speed at an early age compared to frontal and parietal connections. And what's interesting from an electrophysiological perspective is that the timescales of neural signal propagation changed across the lifespan. So the brain seems to have to adapt continuously to these changes, and these developmental changes in the brain. And even like when people are 20 years old, the brain development is not done yet. It's not fixed. There's still growth and still changes, which can either be beneficial or not in some cases. And I showed us some large association fibers, and there's many more, the brain is so diverse in terms of what type of connections it has. And so we're definitely continuing the data collection now at the Mayo Clinic to understand differences between, for example, more rapidly developing projection fibers, callosal fibers, or slowly developing cingulum bundle. And we make the data available with publications at a large scale as we go. And so we can learn a lot from these types of large IAG data collection issues, and we've shared the data so other people can look at things that they're interested in as well. Another example, before I go into this demonstration of how to load these data and what BIDS data look like from an electrophysiological perspective, I would like to show one more example of these data in an individual subject, in a particular network of interest. So rather than taking several of these large association fibers, would like to show a little bit more of these indirect connectivity as well. Because one of the things that we found is that when we stimulated the hippocampus, which is behind this area here in red and annotated here in these pairs, and we stimulated the hippocampus, we measure signals in the posterior cingulate that have this waveform. And now the reason why this is interesting and different from what we measured before is that this peak occurs around 200 milliseconds. And so what we have to think about now is 200 milliseconds is much slower than the 20 to 40 milliseconds that I just showed you before for the direct connections. So this cannot necessarily be related to a direct connection between these two. So we have to use, in order to understand what's generating these types of waveforms, you can see it's reliable on an individual trial basis. In black here, you see the average. In gray, you see the individual trials. This is intracranial data. It's nice to look at because you have individual trial reliability. It was very robust. It's very robust across subjects. We even looked at previous literature, and there's 2 other papers that show this waveform, but don't mention it because people were only looking at this first 20 to 50 milliseconds or so. And so when we bring in diffusion imaging, and we're thinking about when we stimulate the hippocampus, how can the signal spread? As I showed initially, it could go through direct connections, maybe to the posterior cingulate and those regions, but it could also go to indirect connections through the fornix and the cingulum bundle. And so in order to understand better where it goes through, we first have to think about how do we quantify this response? This response has a weird latency around 200 milliseconds. None of our other techniques work. And so Kai Miller yesterday talked about the canonical response parameterization, which basically relies on the cross projection across trials. And since we have these individual trials with these large signals, we can use this technique to think about whether there is a reliable response or not, independent of the duration, whether it's 20 milliseconds, 40 milliseconds, or 200 milliseconds in duration. Because we have these projection profiles and we can basically identify the responses for the duration where we see them. And so we can parameterize the trials and then use the statistics of arbitrary waveforms and arbitrary shapes to look at these different waveforms. So I know Kai talked about this yesterday extensively, and so I just want to point you to some Python code that implements this as well. These are some links to the toolbox where you can parameterize arbitrary waveforms from evoked potentials. And these are some collaborators who implemented the original MATLAB code in Python. And so when we do this canonical response parameterization, we don't have any prior assumptions about latency, polarity, and form. And we can actually see that these hippocampal to posterior cingulate connections are, for example, stronger than amygdala to posterior cingulate connections. So it's more likely that this waveform is really generated by the hippocampus than by the amygdala. We can also look at the significant evoked potentials and separate those based on the exact location and we can compare, for example, these positive and negative responses. So some electrodes show a positive peak, others electrodes show a negative peak. Does that mean that there's something different going on? No, it actually doesn't, because some of these electrodes record 3 millimeters deeper than other electrodes. And what you expect if you have a little source that's a dipole in a cortical, in basically the cortical ribbon, you actually expect that the more superficial electrodes, if you see here in blue, have the opposite waveform compared to the deeper electrodes here in yellow. So this detailed type of response identification allows us to separate these out. And we know that there is a sink and a source between these, or a source between these 2 electrodes, and there's a dipole generating this. So we're not measuring something with these posterior cingulate electrodes that spreads from a different place. We don't have volume conduction. We actually have a very local source that generates these signals. And if we were just looking for a positive peak in the signal, we would have neglected the other half of the responses. So you really, in some cases, when dealing with these data, you need some more free techniques, data-driven techniques that are not where we don't identify exactly what type of response we should be seeing in the data. And there are some other notes on the pre-processing techniques that I'm re-referencing that I'm happy to discuss afterwards. Now, the other thing that we would expect from these data is that if you trace a network and we think that the signal spread from the posterior hippocampus or from the anterior hippocampus to the posterior hippocampus. The other thing you would expect is that when you stimulate further along in a network, you should have an earlier response. And that's exactly what you see here. So when we stimulate in the red sides, in the anterior hippocampus, we see this response here in red. When you stimulate in these green sides, you can see these earlier responses in green. So we think that the red stimulation has a longer way to travel compared to the green sides. And so when we're starting to trace the network, we think that things go from the posterior to the anterior hippocampus. And then we had another 2 patients, and then somehow end up here in this endpoint. And in order to better understand whether it really could travel through these networks, we did diffusion imaging in 2 patients. All data are shared in BIDS. For any questions, you can ask Gabby, who's here in the audience, and luckily got into the Neurohack Academy this week. And then and all the pre-processing was done with QSI prep, which is again based on the data, the diffusion imaging data being stored in BIDS. So we can just use one of these tools to pre-process the data. And the analysis were done in DSI Studio, and specifically because we wanted to obtain all the separate cingulum bundles. There's other packages that identify the cingulum bundle as well, but not in as many subtracts. So we had to use a package that basically separated out these different cingulum bundles. For example, the one that connects the hippocampus to the precuneus versus the one that goes that has the versus these different yellow and blue blue subsegments of the cingulum bundle. And then one of the things that, since this is a course on data science, one of the things you should always worry about when you're dealing with multiple packages and multiple systems is that you have to get all the data into the same space. Because we have all the electrodes in an ACPC framework with the individual data, but then you have the diffusion imaging data pre-processed with QSI prep, and they're in a slightly different, also ACPC-oriented frame. So we have to move the electrodes to the diffusion imaging space. So it's really necessary to always think about where are the electrodes, where are the BIDS derivatives, how do we capture these workflows in a systematic way such that we know which space our analyses are in. Even if you were to be combining, like say, fMRI prep, QSI prep, and the data matching across the two, and you want to stay in single subject space and not go to an MNI space, you have to still worry about which space are my measurements in and how do I integrate those. So in this case, we move our electrodes positions to the QSI prep space, and then we have everything integrated. And what we did in that case is we were lucky enough that some of the electrodes were placed in the anterior nucleus of the thalamus, because in some cases that can help the patient's stimulation there can help can treat the epilepsy, and the neurologists are testing that. And so first we measure this response. We stimulate the hippocampus. We see that this electrode in the posterior cingulum is placed on one of the endpoints of this blue segment of the cingulum bundle, not on the red segment. And that's important because then it could be that the signal spreads through this fornix, mammillary bodies, mammillothalamic tract to the anterior nucleus of the thalamus through the cingulum bundle here, rather than directly to this area. And so when we stimulated the anterior nucleus of the thalamus in this patient, what we also found is that we see the not the exact, but a very similar waveform, but 40 milliseconds earlier. So when we trace this network, use diffusion imaging to confirm that we could potentially follow this, and this is a likely endpoint, we can actually see that this is a likely path for these signals to travel. And so we call this the limbic hap wave, because it's we see it in every patient, and it's such a unique thing from hippocampus to the anterior nucleus of the thalamus to the posterior cingulate. And we hypothesize that it travels this pathway through the fornix, through the cingulum bundle, here to the back of the brain. And so not all of these evoked potentials are related to direct connections. Some indirect connections have strong evoked potentials, and perhaps this is related to the network excitability, and some of these thalamic nuclei having widespread projections, which is interesting because those are things we generally don't consider when we look at cortical-cortical connectivity. And the analysis technique should be suitable for the measured responses. And in some cases, you can use these naive data-driven parameterization techniques to identify new waveforms. So for the scientific portion, before I'll go into the demo, what I've shown is that the transmission speed shows a long developmental trajectory that matches neuroimaging findings. And speeds increase more than twofold from childhood to adulthood. These are large changes. We're not like looking at smaller things, small little changes. And we think that those are driven by microstructural changes in the white matter, such as changes in myelin, and which correlates with axon diameter changes, which both develop for a long time in humans. Stimulation evoked responses, we propose that these may also be used as biomarkers for brain circuits, because in some cases, these waves propagate through many different systems and may be indicative of the integrity of the system. People have lesions or diseases that affect that system. That would be interesting. And the integration of intracranial EEG with imaging helps us understand direct and indirect pathways of neural signal propagation. If you see two areas correlated in fMRI, you can browse some of these data to see if there's actually a direct connection or what the connectivity between those areas actually looks like when you would browse and go back to these shared data sets and look at the intracranial data when we stimulate. What are the latencies? What are the waveforms? What are the evoked potentials? What are potentially spectral changes when you stimulate one area and look in a different region? And so, because I think that these can be used in conjunction with many of these other techniques, I would like to show you some of these data, how to load those in BIDs, and do a demo of that, of those data, so that they can be explored. And of course, we've shared all the data de-identified in BIDs and code on GitHub with all the results that I showed today. And please contact me or post a GitHub issue if you have any questions. So for the, I'm going to thank my lab first before I go to the demo, because otherwise I forget about that. This is the lab at Mayo Clinic. These are some of our wonderful collaborators from a scientific and clinical perspective. And this is Mayo Clinic in the summer in Rochester. It's wonderful. In the winter, we do other fun stuff like beat a record on an escape room and those types of things. Okay, sorry, I got kicked out of everything when I got out of my presentation, and now when switching to the demo.
Dora, don't forget to share your screen.
Yeah. Share. Okay. 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 Can people see my screen? Thumbs up? Yeah. Racing, I think. On Zoom. Okay. We're back up and running. So if we look at these types of intracranial data, these are quite unique resources. And it's rare. So we spend a lot of time getting these together in BIDs. And they're dependent on being able to run these depends on several Python packages. So we're just going to go to the top of the Jupyter Notebook. This Jupyter Notebook is also shared on the Jupyter Hub. So if you want to walk along, you can open it. And please raise your hand if you have any issues with any portions. I'm happy to pause throughout and see if there's if we can debug anything on the way. So we have the standard Python packages. IEG prep is a package that was written by one of my postdocs to actually can load various types of IEG data and load epochs. So it's quite convenient. So if you would be able to load this and have those packages installed, it'll help you pretty easily actually load either some of the data that are saved in EDF format, some of the data that are saved in brain vision format. For the electrophysiology derivatives in BIDs, we don't just have a nifty format as fMRI does. where there's like over 20 different types of formats that amplifier companies use to save their data. A few of those are accepted in BIDS, and those are all and those can all be loaded using IAG prep. So it's kind of, which is why one of these using that is actually quite convenient. M&E Python also has a lot of options to load these packages or to load these different data types. If you're interested in that, that's another tool that's being actively developed. It can be really useful if you're loading these data. I know Alex Rockhill is maybe be joining part of the course next week. If you have any questions about M&E Python, ask him. He's great at it. I'm loading the BIDS dataset path here. I think on the Jupyter Hub, this is a slightly different path. This is just where I put the I put this dataset that is from OpenNeuro. The dataset on OpenNeuro that we're looking at was the one I showed with development about H, which is this really large 74 subject dataset. It's 300 gigabytes in total. So for this example, we only synced 2 subjects just to be able to show you how this runs and what the data look like. And so I downloaded only 2 subjects in this case. And so this is just a path to wherever your data are located. The BIDS package, import BIDS, here you can see that we're importing, allows you to easily access data however they're shared, whether it's imaging data or electrophysiology data from any source in BIDS. So here I'm running this portion, and you can see that the BIDS layout immediately shows that there's 2 subjects, there's 2 sessions, 2 runs. The subjects in this session is number 33 and number 35, which are just 2 example subjects that I loaded here. If I go back to the folder, if I just show you briefly what this folder looks like, this BIDS folder looks like here, you can see that these are 2 of the subjects that are downloaded. You can see that there's a participants TSV file, dataset description. These are all standard human readable data structures in BIDS. I removed some of the other subjects so that it would run easily and we would be able to get these 2 subjects, but you can find all 74 subjects on OpenNeuro. So you can see here, there's 2 subjects. These are the 2 examples we loaded. You can also see that here within this subject, there's 1 session. Here's the IEEG folder. You can also imagine having an NAT folder, an fMRI folder, an EEG folder, a DMRI folder, DWI folder, whichever data type you have. In this case, this is we're going to look at some of these electrophysiology data types, specifically the .eeg file, which is accompanied by a V header and a V marker file, which basically is the header information related to this file. So let's actually load some of these files in BIDS, use this BIDS layout to load some of these files, and let's look at the content. And so let's do that for subject number 33. This is the run name, which depends on and then we're going to look at one particular stimulation site. We stimulated in these subjects all the electrodes. They did pair by pair by pair by pair. They're mapping all the connections from all the different areas. That's their protocol that they use at the UMC Utrecht, which is really generates an extremely rich data set, because then you can map basically all connections, any site that was stimulated. We don't have as much time at the Mayo Clinic to map all connections, so we use a subset of selected sites that we're interested in, try to get as many as possible. But just given the time that these things allow, the time that allowed in these studies, we generally get a subset. So we're looking at this first session, the first task, and we're going to retrieve some of the names. For example, the IEG name, the JSON name, which has, for example, the important information like the sampling frequency, the channels names, which has which channels were reported, events, what was happening, did you show a visual stimulus, electrostimulation, this is an electrostimulation data set, and the electrodes, which has information on where the electrodes were placed. And in this data set, the electrodes are all converted to MNI space for easy accessibility, and because it's also, that was the only thing we were allowed to share. We were not allowed to share the individual subject space data, because then you could potentially know the size of an individual brain. So while we do, and it could potentially be traceable back to the individual subject. So for de-identification reasons, we are only allowed to share the MNI space brains. And you can see that all these data exist, they're successfully loaded. We have electrode descriptions, events names, etc. And then we use the pandas data frame function to quickly test what the expected function have, what the files, what type of information the files have. So let's look at the channels files. You see that the channel names were CO1, CO2. These are ECOG channels, which is an electrocorticography channel placed on the brain surface. There's units or microvolts. They're filtered with a low pass and a high pass filter by the hardware we're reporting. These are grid electrodes. The sampling frequency was 2000 Hertz. There was no notch filter applied. In the status, we often say whether channel is good or bad, because some channels have noise and then we mark them as bad, which is extremely helpful if you're looking at these data afterwards. All these data are actually extremely well annotated. So we know which channels were measured. And then let's look at events. And we look at events number 30 to 40 here, because in the beginning, if we look at the first events, those are just artifacts that were annotated. But from 30 to 40, we actually see we did stimulation. Electrostimulation was applied in this case for 1 millisecond, which is longer than what we're allowed to do at Mayo Clinic. The onset is noted here as well. The pair that was stimulated, so we stimulated the pair, for example, electrode pair CO2, CO3, CO3, CO4, etc. You can see that there was an electrostimulation current of 8 milliamps. So this has all the important information that we need in order to understand what was done. Then we can look at the electrode positions. These are XYZ positions in MNI space. Now, this is important when you want to integrate your data with any other type, because these are just standardized MNI 305 positions. And you can see here that there's also, just for easy access, there's a label added from the Destrier Atlas. So you can see that these electrodes, for example, in the middle, in the middle occipital gyrus or the middle temporal gyrus, all these labels are added for easy access. But since they are in MNI space, you would be able to see that as well. Now we can render the brain surface. And this is, for me, this actually opens in a separate window. For everybody here on the Jupyter Hub, this is actually saved in a viewer.html page next to the Jupyter notebook. So this is a Jupyter notebook I'm looking at, and this is actually saved. You can double click on that HTML file and it'll open in the browser, and you can see where the electrodes were placed. Now, there's a couple of things that are important to note. One of those is that these electrodes do not seem like they're spaced in a grid, but that's because they're placed in MNI space. And all these electrodes are normalized to MNI space using the free surfer segmentation. So they're snapped to the closest portion on the free surfer. Free surfer brain is inflated to a sphere and then morph back into MNI space, such that you actually get, such that an electrode that is placed on the precentral gyrus is not suddenly going to end up on the postcentral gyrus. So this is actually the fact that these are distorted provides a more accurate MNI space location compared to if we use something like an SPM co-registration, which maintains the grid configurations a little bit better, but can move electrodes from gyrus to gyrus. So it's actually, so you can be pretty confident that every electrode, the gyrus on which it's positioned in MNI space, is actually the gyrus where it was measuring in the individual subject. So for example, these electrodes here on the pre and post-central gyrus are actually located there. And so then we can, after we know which electrodes we're at, we can look at, we can actually load some of the data. And we can, for example, we can load some of the IEG data, which is here that's the IEG file name. And we can look at some of these stimulation sites. So let's look at this stimulation site that we identified here on the top for this subject, CO3, CO4. You can see here that CO3, CO4, CO1 is right there. So when we go down to CO2, CO3, CO4, we're stimulating some of these parietal regions. And we can see, for example, what happens when we stimulate those parietal regions. Let's first load some of those data. And we can see that what we're loading, if we go just right underneath this structure, we load some epochs. Those epochs have the shape of 133 by 10 by 6,000 something. So there's 133 channels where responses are measured. Electrodes were stimulated 10 times. So there's 10 stimulations. This is the time. These are the time points. So let's plot from one of the electrodes. There's one of these 133 electrodes. Let's pick electrode 10 and just plot all the trials just to see what the data actually look like for one of these. And if we do that, you can see for this electrode, and of course I picked one with a response, you can see that there's this giant artifact at around 0 milliseconds, which is stimulation related. So that's a stimulation related artifact. Every time you do electrical stimulation, you're going to saturate an amplifier, you're going to generate this artifact. But after this artifact is over, in this case, after 10 milliseconds, you can see these beautiful responses. And these are single trials. So on every single trial, you can actually see this negative potential, which is what we quantified to study the latencies. You can see that these data are extremely reliable and nice to work with when they're this clean. And so this is just showing the data from, for example, electrode number 10. But if we want to look at all the other electrodes, we can visualize that in different ways just to check our data. And we can look at, these are all the channel names, the channel indices from the good channels. You can see here that I selected only the good channels, and I'm plotting the responses as a function of time here for all the channels. And we can see that there's many channels that have a good response when we're stimulating this site. So this site has a lot of outgoing connections, strong connectivity to many other regions. And you can actually look at that and compare that to different channels that are close by, like what are the hubs that are actually driving a lot of other areas. And so this is just, this is pretty basic. I'm not doing any advanced connectivity analyses, but that's because this is what you actually need to look at before you do that, because you need to look at the single trials, look at whether there's outliers, and look at whether all the channels are looking good, and do a thorough data visualization before starting to throw any more advanced tools at these data. And so I thought that this would be the most helpful Jupyter notebook to kind of share if you're interested in combining these with anything else for any purposes, for example. So I'm happy to take any questions.
It's a question. Thanks. That was beautiful. Really cool link of everything, like starting from bits to the book academies. What are the key hurdles that you had with the BITS organization so far? And how do you see that project? Like, there's always this scoping problem that is always in our mind, like, what's how far do we go into the details, because those are useful? How far do we limit those things? Like, how do you see the bits standardization going, and where is it going?
Okay, so yeah.
Thoughts on the general problem.
So in terms of where BITS is going, like, is it going to go more into detail for specific things or more broader expand, basically? And I think that that's one of the things that will be driven by the community. It will be driven by the needs of the students. What do people who are working with the data actually need? In our case, there's there are some more in-depth things that are needed. For example, in terms of labeling, like quantifying our workflows, which is what you're working on, which is wonderful, which actually, like, how do we actually capture these workflows so that we can do them reliable and share them, especially for these multimodal integrations? How do we share that type of work such that other people can do that more easily? So there's some of that, which is largely driven. There are some details in terms of how do we actually annotate each event such that people can understand exactly what we did. What's an artifact? Like, am I going to call that bad, an artifact? such that if someone loads the data, they're not accidentally going to, like, say, the subject had large interictal activity. We annotated that in this case in the data. Here you can see in the events file that the first set of data all has artifact labels on it. Someone might just not look at that and not know that that was actually annotated, that from around these time for this duration, there was an artifact in the data. We don't have a standard for this. We just threw it in the events.tsv file. So we basically overload some of these files without a structure. I think there'll be some efforts in BIDS to standardize those further. I think so it's basically driven by what people need, right? Yeah.
On that specific problem, I think it's a even more difficult problem than it seems, because at least for fMRI, some of those bad or good will be entirely project dependent.
Yes.
So you can't actually put that as attached to the data. It has to be attached to the project in some study.
That's also the case here, because for example, the interictal activity is an artifact for us. So we say that's bad. For the neurologists, that's actually what they're interested in.
Right.
So it really depends on the question that is being asked. And in this case, this is just called artifact, but it could be anything between an electrical artifact, someone moving the channels around, or interictal discharges. And we don't actually know the difference now that we look back at the data, because there's no, So those types of things, I think, are going to make the data that are already there, enrich the data that are already there. Some people are, for example, working with eye tracking data or motion capture data and expanding that, because there's a community need for that, and people want to integrate that with their data in BIDS. And I think that those are a lot of the, so wherever the needs are, that's where things are going to be driven. It's very much, we don't impose a lot of things from the steering committee in terms of where people should be going. It really depends on what people actually need. And so it's very practical, which I like. It's not very, we don't decide. It's just like, what's the most practical solution? What actually works?
I think there's a question on Zoom. Ashley, do you want to unmute and?
Hi there. Thanks so much for the presentation. I was just curious about this process right now. When you do those annotations, is it a manual process or could it be automated?
So it depends on what it's currently a combination of both. So I like reviewing all the data still, clicking through it and noting any large artifacts. So we still do that in the lab a lot. Just because it's good to look through all your data and just see what's in there. So I make anyone who joins the lab, any PhD student, they have to look at the raw data and see what's coming in. Because otherwise, if you've never looked at the raw data, you really don't know what you're dealing with. And but then they can write an automated artifact detector. And if that works well and doesn't get rid of too many trials, still keeps most of the data, then they can also use an automated tool. But these data are so variable that it's really difficult to have a completely automated tool. So I think it will always be a combination between the two. Bad channels is still something that's relatively easy to detect. We just mark it. One person can basically do it for the lab. We know that it's going to be bad across all the sessions. So it's a combination between automated and manual marking, but that's just because of the data type. I think there's more automated tools for MRI for detecting bad slices, but you still have to set a threshold and decide, like, what are you going to do with that slice? If you have a bad slice in your data, it's occurring in one scan. Are you going to get rid of the whole scan? Are you going to interpolate it based on the neighboring slides? It just depends on the project, what the best approach is, but we are basically, we do a combination of those.
Awesome, thank you.
Thank you.
Thanks so much to Dora.